The cochlea/kk.li/, ancient Greek meaning spiral or snail shell, is the auditory portion of the inner ear. It is a spiral-shaped cavity in the bony labyrinth, in humans making 2.5 turns around its axis, the modiolus. A core component of the cochlea is the organ of Corti, the sensory organ of hearing, which is distributed along the partition separating fluid chambers in the coiled tapered tube of the cochlea. The name is derived from the Latin word for snail shell, which in turn is from the Greek kappa omicron chi lambda alpha cochleus, snail, screw, from kappa chi lambda omicron cochleus, spiral shell. In reference to its coiled shape, the cochlea is coiled in mammals with the exception of monotremes. Structure. The cochlea, plural is cochleae, is a spiraled, hollow, conical chamber of bone, in which waves propagate from the base, near the middle ear and the oval window, to the apex, the top or center of the spiral. Rosenthal's canal or the spiral canal of the cochlea is a section of the bony labyrinth of the inner ear that is approximately 30 mm long and makes 2/3 turns about the modiolus. The cochlear structures include 3 scalae or chambers. The vestibular duct or scala vestibuli, containing perilymph, which lies superior to the cochlear duct and abuts the oval window. The tympanic duct or scala tympani, containing perilymph, which lies inferior to the cochlear duct and terminates at the round window. The cochlear duct or scala media, containing endolymph, a region of high potassium ion concentration that the stereocilia of the hair cells project into. The helicotrema, the location where the tympanic duct and the vestibular duct merge, at the apex of the cochlea. Reissner's membrane, which separates the vestibular duct from the cochlear duct. The basilar membrane, a main structural element that separates the cochlear duct from the tympanic duct and determines the mechanical wave propagation properties of the cochlear partition. The organ of Corti, the sensory epithelium, a cellular layer on the basilar membrane, in which sensory hair cells are powered by the potential difference between the perilymph and the endolymph. Hair cells, sensory cells in the organ of Corti, topped with hair-like structures called stereocilia. The cochlea is a portion of the inner ear that looks like a snail shell. Cochlea is Greek for snail. The cochlea receives sound in the form of vibrations, which cause the stereocilia to move. The stereocilia then convert these vibrations into nerve impulses which are taken up to the brain to be interpreted. 2 of the 3 fluid sections are canals and the third is a sensitive organ of Corti which detects pressure impulses which travel along the auditory nerve to the brain. The 2 canals are called the vestibular canal and the tympanic canal. Detailed anatomy. The walls of the hollow cochlea are made of bone, with a thin, delicate lining of epithelial tissue. This coiled tube is divided through most of its length by an inner membranous partition. 2 fluid-filled outer spaces, ducts or scalae, are formed by this dividing membrane. At the top of the snail shell-like coiling tubes, there is a reversal of the direction of the fluid, thus changing the vestibular duct to the tympanic duct. This area is called the helicotrema. This continuation at the helicotrema allows fluid being pushed into the vestibular duct by the oval window to move back out via movement in the tympanic duct and deflection of the round window. Since the fluid is nearly incompressible and the bony walls are rigid, it is essential for the conserved fluid volume to exit somewhere. The lengthwise partition that divides most of the cochlea is itself a fluid-filled tube, the third duct. This central column is called the cochlear duct. Its fluid, endolymph, also contains electrolytes and proteins, but is chemically quite different from perilymph. Whereas the perilymph is rich in sodium ions, the endolymph is rich in potassium ions, which produces an ionic, electrical potential. The hair cells are arranged in 4 rows in the organ of Corti along the entire length of the cochlear coil. 3 rows consist of outer hair cells, oaks, and one row consists of inner hair cells, IHCs. The inner hair cells provide the main neural output of the cochlea. The outer hair cells, instead, mainly receive neural input from the brain, which influences their motility as part of the cochlea's mechanical preamplifier. The input to the OC is from the olivary body via the medial olivocochlear bundle. The cochlear duct is almost as complex on its own as the ear itself. The cochlear duct is bounded on three sides by the basilar membrane, the stria vascularis, and Reissner's membrane. Stria vascularis is a rich bed of capillaries and secretory cells. Reissner's membrane is a thin membrane that separates endolymph from perilymph, and the basilar membrane is a mechanically somewhat stiff membrane, supporting the receptor organ for hearing, the organ of Corti, and determines the mechanical wave propagation properties of the cochlear system. Function. The cochlea is filled with a watery liquid, the perilymph, which moves in response to the vibrations coming from the middle ear via the oval window. As the fluid moves, the cochlear partition, basilar membrane and organ of Corti, moves. Thousands of hair cells sense the motion via their stereocilia, and convert that motion to electrical signals that are communicated via neurotransmitters to many thousands of nerve cells. These primary auditory neurons transform the signals into electrochemical impulses known as action potentials, which travel along the auditory nerve to structures in the brainstem for further processing. Detailed events. The stapes, stirrup, ossicle bone of the middle ear transmits vibrations to the fenestra ovalis, oval window, on the outside of the cochlea, which vibrates the perilymph in the vestibular duct, upper chamber of the cochlea. The ossicles are essential for efficient coupling of sound waves into the cochlea, since the cochlea environment is a fluid membrane system, and it takes more pressure to move sound through fluid membrane waves than it does through air. A pressure increase is achieved by the area ratio of the tympanic membrane to the oval window, resulting in a pressure gain of about 20 times from the original sound wave pressure in air. This gain is a form of impedance matching to match the sound wave traveling through air to that traveling in the fluid membrane system. At the base of the cochlea, each duct ends in a membranous portal that faces the middle ear cavity. The vestibular duct ends at the oval window, where the footplate of the stapes sits. The footplate vibrates when the pressure is transmitted via the ossicular chain. The wave in the perilymph moves away from the footplate and towards the helicotrema. Since those fluid waves move the cochlear partition that separates the ducts up and down, the waves have a corresponding symmetric part in perilymph of the tympanic duct, which ends at the round window, bulging out when the oval window bulges in. The perilymph in the vestibular duct and the endolymph in the cochlear duct act mechanically as a single duct, being kept apart only by the very thin Reissner's membrane. The vibrations of the endolymph in the cochlear duct displace the basilar membrane in a pattern that peaks a distance from the oval window depending upon the sound wave frequency. The organ of Corti vibrates due to outer hair cells further amplifying these vibrations. Inner hair cells are then displaced by the vibrations in the fluid, and depolaris by an influx of K and via their tip-link connected channels, and send their signals via neurotransmitter to the primary auditory neurons of the spiral ganglion. The hair cells in the organ of Corti are tuned to certain sound frequencies by way of their location in the cochlea, due to the degree of stiffness in the basilar membrane. This stiffness is due to, among other things, the thickness and width of the basilar membrane, which along the length of the cochlea is stiffest nearest its beginning at the oval window, where the stapes introduces the vibrations coming from the eardrum. Since its stiffness is high there, it allows only high-frequency vibrations to move the basilar membrane, and thus the hair cells. The farther a wave travels towards the cochlea's apex, the helicotrema, the less stiff the basilar membrane is, thus lower frequencies travel down the tube, and the less stiff membrane is moved most easily by them where the reduced stiffness allows. That is, as the basilar membrane gets less and less stiff, waves slow down and it responds better to lower frequencies. In addition, in mammals, the cochlea is coiled, which has been shown to enhance low-frequency vibrations as they travel through the fluid-filled coil. This spatial arrangement of sound reception is referred to as tonotopy. For very low frequencies, below 20 Hz, the waves propagate along the complete route of the cochlea differentially up vestibular duct and tympanic duct all the way to the helicotrema. Frequencies this low still activate the organ of Corti to some extent, but are too low to elicit the perception of a pitch. Higher frequencies do not propagate to the helicotrema, due to the stiffness-mediated tonotopy. A very strong movement of the basilar membrane due to very loud noise may cause hair cells to die. This is a common cause of partial hearing loss and is the reason why users of firearms or heavy machinery often wear earmuffs or earplugs. Amplification by the hair cells. Not only does the cochlea receive sound, it generates and amplifies sound when it is healthy. Where the organism needs a mechanism to hear very faint sounds, the cochlea amplifies by the reverse transduction of the oaks, converting electrical signals back to mechanical in a positive feedback configuration. The oaks have a protein motor called prestin on their outer membranes. It generates additional movement that couples back to the fluid membrane wave. This active amplifier is essential in the ear's ability to amplify weak sounds. The active amplifier also leads to the phenomenon of sound wave vibrations being emitted from the cochlea back into the ear canal through the middle ear, otoacoustic emissions. Otoacoustic emissions. Otoacoustic emissions are due to a wave exiting the cochlea via the oval window, and propagating back through the middle ear to the eardrum, and out the ear canal, where it can be picked up by a microphone. Otoacoustic emissions are important in some types of tests for hearing impairment, since they are present when the cochlea is working well, and less so when it is suffering from loss of oak activity. Role of gap junctions. Gap junction proteins, called connexins, expressed in the cochlea play an important role in auditory functioning. Mutations in gap junction genes have been found to cause syndromic and non-syndromic deafness. Certain connexins, including connexin 30 and connexin 26, are prevalent in the two distinct gap junction systems found in the cochlea. The epithelial cell gap junction network couples non-sensory epithelial cells, while the connective tissue gap junction network couples connective tissue cells. Gap junction channels recycle potassium ions back to the endolymph after mechanotransduction in hair cells. Importantly, gap junction channels are found between cochlear supporting cells, but not auditory hair cells. Clinical significance. Bionics. In 2009, engineers at the Massachusetts Institute of Technology created an electronic chip that can quickly analyze a very large range of radio frequencies while using only a fraction of the power needed for existing technologies. Its design specifically mimics a cochlea. In other animals, the coiled form of cochlea is unique to mammals. In birds and in other non-mammalian vertebrates, the compartment containing the sensory cells for hearing is occasionally also called cochlea, despite not being coiled up. Instead, it forms a blind-ended tube, also called the cochlear duct. This difference apparently evolved in parallel with the differences in frequency range of hearing between mammals and non-mammalian vertebrates. The superior frequency range in mammals is partly due to their unique mechanism of preamplification of sound by active cell body vibrations of outer hair cells. Frequency resolution is, however, not better in mammals than in most lizards and birds, but the upper frequency limit is sometimes much higher. Most bird species do not hear above 4-5 kHz, the currently known maximum being 11 kHz in the barn owl. Some marine mammals hear up to 200 kHz. A long coiled compartment, rather than a short and straight one, provides more space for additional octaves of hearing range, and has made possible some of the highly derived behaviors involving mammalian hearing. As the study of the cochlea should fundamentally be focused at the level of hair cells, it is important to note the anatomical and physiological differences between the hair cells of various species. In birds, for instance, instead of outer and inner hair cells, there are tall and short hair cells. There are several similarities of note in regard to this comparative data. For one, the tall hair cell is very similar in function to that of the inner hair cell, and the short hair cell, lacking afferent auditory nerve fiber innervation, resembles the outer hair cell. One unavoidable difference, however, is that while all hair cells are attached to a tectorial membrane in birds, only the outer hair cells are attached to the tectorial membrane in mammals.
Please subscribe and thanks for watching.